Hi all, Dr. Clark here. Today we're going to talk about how enzymes work. Okay? And so we left off talking about how things flow through a system, a little bit about energy. Okay? We'll get in much more detail with ATP and stuff like that in just a little bit. But we need to talk about how enzymes function and talk about biochemical pathways. This is a biochemical pathway here, and you can see that there is a lot of enzymes associated with this biological pathway for MORT. Okay, what MORT does is it can regulate translation, metabolism, things like that. So it has a link to things like insulin receptors, growth factors. There's all kinds of things. So we'll look at not nearly as complex, but I just wanted to show you that when I talk about biochemical pathways, it's not just like 4 proteins in a row. I mean, literally, it can be a massive amount of proteins, and depending on what is present in the system, depends on what route that molecule will take. And we'll come back to this. All right, so talk a little bit more about enzymes. So remember when I was talking about 2 types of chemical reactions. You have endergonic reactions where you have to supply energy, and exergonic reactions where more energy is released. So endergonic, you supply more energy, but the end product has more energy than the reactants. Exergonic, you might have to supply a little bit of energy, but the end product has less energy than the reactants. Therefore, exergonic releases energy, endergonic consumes energy. Well, in order to mediate or decrease the amount of energy needed for a reaction to occur, you have to reduce the activation energy. And one way to do that is by using an enzyme. Okay? So enzymes catalyze reactions, and most of you know, and you've probably heard this before, enzymes speed up reactions. Well, they do so by reducing how much energy is needed for the reaction to occur. Okay? The other thing that enzymes have that most of you already know, because we've kind of talked about this in different situations in lab and things like that, is enzymes are specific proteins. And specific meaning they only bind to typically one substrate or a couple substrates to make a single product. But it's the same enzyme, same substrate combination always. For example, if we want to break down sucrose, which is table sugar, we'd use the enzyme sucrase. If you want to break down hydrogen peroxide, which is a byproduct of cellular respiration and is poisonous to us, you would use the enzyme catalase. If you want to be able to drink milk and break down milk sugars, which we call lactose, you would need the enzyme lactase. And on and on and on and on, okay? And they're very specific. You cannot use lactase to break down sucrose. Okay? It doesn't work that way. Okay? On these enzymes, they'll have what we call an active site, or sometimes called an activation site, okay? Which is an area at which the substrate will bind. And you might have multiple substrates, and therefore you might have multiple activation sites or active sites. So they're going to bind to the surface. A lot of times it occurs in kind of a groove that the molecule can fit in there, and then the protein can change its shape to fit that molecule. Okay. The other thing that you can have is you can have a binding site. Okay. Sometimes the active site and the binding site are the same. Okay. Other times you have an active site and then another site on the molecule, which is a binding site, and it could be a regulatory binding site. Therefore, a molecule must fit in there to open the active site, or a molecule would fit in there to close the active site. Okay? And we'll talk about these a little bit later when we talk about how we regulate enzymes. So here you can just see kind of a diagram. Remember, proteins are just amino acids linked together with peptide bonds. So all these little balls actually are, could represent the amino acid. Okay? And so you get all these amino acids linked together in some kind of globular form. Remember, their primary form will be a long chain, and then secondary they'd wrap, and then tertiary they'd wrap again. Quaternary they'd wrap with other tertiary proteins. So, this is just hypothetically like, what it would look like. You have this active site or activation site, and a reactant would fit in there. When it fits in there, the protein changes shape. Now, I want to make sure that you understand what's going on here, okay, with this protein changing shape. This is called an induced fit. Okay, so the protein changes shape to fit this molecule. And a lot of people, when they hear that, they're like, well, are proteins alive? Do they have muscle movement? What's going on here? Okay? They're not alive. I mean, proteins are building blocks of life, but they're not alive themselves. What's going on is it's a chemical reaction. So this reactant is going to probably, it doesn't have to be polar, but often it'll have, maybe a positive side and a negative side. It'll often be polar. And then the protein itself will be polar, maybe in the opposite way. So they're attracted to each other. So they pull together to each other. And other times it'd just be this material having maybe just a simple hydrogen bond with this material. Okay, so a weak force, a weak bond, attraction to each other, and that's kind of driving that protein to wrap around it. Okay? In doing so, when the protein wraps around it, it often will change the shape of the reactant. Okay? Just a little. Okay? Just enough to open up a weak bond, right? So water can come in and you can get a hydrolysis reaction. Okay? So remember, you have dehydrogenous reactions where water is formed from the reaction. Two reactants come together, water is formed. And then you have dehydrogenous reactions where water is used to break something up. In this case, since we only have one reactant, it's probably going to be split into two products. Okay. Now down here, these might indicate a binding site, or what we sometimes call an allosteric site, okay, a site where this right here can be controlled by this site. Okay, we'll come back and look at those. Okay, so again, it's not just that one substrate fits one enzyme. Okay? It's that one substrate will fit in and then an induced fit will occur. Okay? This allows for the protein, the enzyme, to really match the structure of the substrate or substrates. Okay? And so that's really what makes it so enzymes are not usable for other functions. Okay? Because they only will have one induced fit with one or maybe a couple molecules, and that's it. Okay? Again, how do they work? They lower the activation energy needed to either form a bond or break a bond. Okay? And to do this, like I said before, they change the shape of the substrate so that more water can enter, or so it weakens the bonds so the two bonds can form together if you're going to form a product from two substrates. Okay, and we'll look at some videos of this so you can get an idea of what I'm talking about. But that induced fit, okay, the change the shape of the substrate is what's going to reduce that activation energy. The amount of energy needed for a product to form is going to be reduced. Okay, that's how enzymes work. So yes, they speed up reactions, but in some cases, without the enzyme, the reaction would never occur. Okay? So it's not just a matter of speeding up the reaction. It's a matter of reducing how much energy is needed. In some reactions, there's so much energy that would be needed for that reaction to occur spontaneously, okay, that an enzyme has to be used. Okay. The other thing is most of you know, because you've done labs on this, or we've talked about it before, enzymes are reusable. This is an amazing evolutionary mutation to allow for enzymes to be reusable. Because think about it if they weren't. Okay? Let's say you eat a spoonful of sugar. Okay? It would go down to your gut, sucrase, that enzyme would, break up sucrose into glucose and fructose. Okay? But then if that enzyme was dissolved, then you would have to wait a period of time for that enzyme to be remade. And that would mean, like, literally, your DNA would have to turn back on, copy your DNA into a messenger RNA, make the protein would have to fold, okay, and then be redistributed into that region. This takes time, lots of time. So you might be able to eat if this was the case, you might be able to eat one spoonful of sugar. The next spoonful of sugar that you were to take, you might get no nutrients from it. Okay? It would just come out. And so this quirk of evolution has really allowed organisms to survive. Okay? And without it, really no organism would be able to survive if they had to make enzymes every time they use them. So here's that process again. Some people like to think of it as kind of a lock and a key, okay, or two keys and one lock. It just depends on what you're dealing with. But here you can see, in this case, there's an active site that fits two substrates. The two substrates would come in. Okay. This enzyme is going to have an induced fit. It's going to change the shape and change the bonding strength of these two substrates. By doing so, these material will bond with each other and form a single product. And then the enzyme snaps back to its original shape.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
Okay, so again, like we stated before, that weakens the bond, forms a product, or there are enzymes that would take this product, weaken the bond, and make two substrates. And we're going to look at one of those right now.
An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
Okay, so that brings us to those three last points. Okay? So this can occur over and over again. Remember, enzymes are reusable until they become denatured. So we're going to go through how to denature a protein. Some of you already know this. Okay? Inhibited, so is there an allosteric site or is there a competitor molecule that would stop this from occurring? Okay? Or we're not really going to cover just wears out, but over a long period of time, your enzyme the bonds will just fall apart, okay? And you'll eventually need to make a new enzyme because it's just old. Okay? All right, before we get into kind of how we can denature enzymes, we're going to talk about biochemical pathways. And this is a way at which we can control products that are formed from substrates. Okay? A biochemical pathway, right, is a series of proteins, series of enzymes that occur along a cell membrane, okay, that the product of one of the enzymes is the substrate for the other enzyme. Okay? And on and on and on. Okay? So you have a substrate that comes in, creates a product. That product is now the substrate for the next enzyme. On and on and on until a final product is produced. Okay? These are called biochemical pathways. Okay? Now, we often show you simple things like, this diagram here is fairly simple. 4 enzymes, 1 product. But there are lots of situations, especially when we start talking about photosynthesis and cellular respiration. There'll be lots of situations where I say, oh yeah, and there is a 12 protein biochemical pathway, or 10 protein biochemical pathway. And that's what I'm referring to. That means that you have 10 proteins in a row or 12 proteins in a row, and one substrate comes in, products form, next substrate, blah, 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 all the way to the end product. Okay? All right, so again, we can talk about things that would affect enzyme activity. And we've already talked about this a little bit. You've had a lab on it. Okay? And that is temperature and pH affect enzyme activity. Okay? And what I mean by that is you can denature or you can break up a protein, okay, by increasing the temperature above its optimum temperature range. Okay? You can also destroy a protein by changing the pH or the optimal pH range. Okay? Now the thing about temperature is you have to increase temperature to get a change in the protein. Okay? You're going to unfold it, it's going to refold, but it's not going to fold in the same way, and it'll be denatured and broken. Okay? Enzyme function, when it comes to optimal pH, it just depends on what the optimal pH range is. So if the enzyme is occurring in the blood of an organism, okay, then the pH range is 6 to 8. Okay? And if you increase it too much, you'll denature it. If you decrease the pH too much, you'll denature it. So you can't go either direction. Okay? As far as things like stomach enzymes, where the pH of the stomach might be 2, it might have a pH range of 1 to 3, okay? And it's hard to denature it going down the acid. So going more acidic, you're not going to really denature it. You might cause some issues, but you're not going to denature the protein. But going the opposite way, going up the pH scale, so becoming more basic, would definitely do that it would damage the pH. Okay? And we'll look at some examples of that here. Okay, so here you can look at temperature. So the optimal temperature for most human enzymes, because we have a constant body temperature, is somewhere around 40 degrees Celsius or 98.6 degrees Fahrenheit. Okay? The optimal temperature for things like enzymes in hot springs, like the Yellowstone hot spring bacteria, okay, might be 70 degrees Celsius, or in some of those hot springs where the water is boiling, you might be all the way to 100 degrees Celsius is their optimal temperature range. And some of the enzymes might have an optimum from like 90 all the way down to 0. They could have or close to 0. They could have a huge span of optimal temperature ranges. Okay? And organisms like reptiles, amphibians, fish, okay, their enzymes have a much wider range of temperature because their body has a much wider range of temperature, and it can tolerate much wider ranges of temperature. Okay. As far as pH goes, okay, even within an organism, so if we look at just humans, we have pepsin in our stomach, okay, and trypsin in our blood. And you can see there, trypsin, its optimum is probably 6.8 or close to the pH of blood. Okay, and then pepsin, its optimal is going to be right around stomach acid, but it has a range. Okay, now if you get to 0, okay, then it's outside the range, and you could start destroying that enzyme. Okay, the pepsin enzyme. All right. So how do we regulate them besides destroying them? Because you don't want to destroy them. Okay? So we want to control enzymes. And you might ask, why do you want to control an enzyme? Well, what if you have an enzyme that's making something or, breaking down proteins? So you consume protein, and you have an enzyme that breaks down that protein. It's going to break that down that protein into amino acids. Okay? Well, what if we're going to use those amino acids to build more proteins? Okay. We don't want to continuously break down proteins and create a bunch of amino acids that we have no use for, especially if we're not building new proteins. Okay, so we want to be able to control the process of breaking down material, okay, or building material. Maybe, in the process of breaking down glucose, you're going to trigger insulin and things like that. Maybe you don't want to burn through all your insulin supply, okay, when you're not doing anything, when you're not active. Okay? So instead, you might want to store that excess sugar as fat, okay? And that's what we do, but it's regulated through enzyme regulation. Okay? And most of the time, it's controlled by what we call an allosteric enzyme, or the enzyme is called an allosteric enzyme because it has an allosteric site. Okay? An allosteric site is a binding site on some other region of the enzyme, okay, away from the active site, that can control the active site. Okay? It can control it by having a repressor come in. So the repressor would come into the allosteric site and it would close off the active site. Or you can have an activator. An activator would come in to the allosteric site and it would open up the active site. Okay? And so here's a diagram to show you what I'm talking about. So you have a substrate that would come straight in to the active site, produce a product. Okay? But you could add a repressor. Sometimes it's a byproduct of the products. Sometimes it is the product that would fit back in. So if you have too much product, sometimes that'll slide back into the allosteric site and shut this down so you don't make any more. Okay? But this repressor would come in, hook up, and then you get, a closed off site. So you get the active site closed and it cannot bind, so no more product can be formed. You get the opposite, okay, where you can get this substrate can't fit until an activator comes in, then it fits, product's made. Okay? A couple different ways that you can have these products form. Okay? This is called feedback inhibition, and you can have negative and positive feedback inhibition, where negative means that basically too much product is being produced, so we need to shut the source down. Positive feedback would be, hey, there's not enough of this, thus increase and turn the system back on. Okay? Two ways at which you can get feedback inhibition from kind of a negative standpoint, where you're going to shut down the system, where you're going to repress the system. You can have competitive inhibition, where you get a molecule that fits into the active site and it blocks the active site. Or you can get non-competitive inhibition, where you have a molecule that fits in the allosteric site and closes off or changes the shape of the active site. Okay. So here you can see how both of these work. So here you have this competitor, okay, that will fit in there, and it's not going to be a great fit, but it's not going to allow that substrate to bind. Okay, so you might get simple hydrogen bonds that are forming around this, so this molecule stays in there and the substrate just cannot bind with it. Okay? Or you can have non-competitive, where you have a molecule that comes into the allosteric site, changes the shape of this, and the substrate doesn't fit anymore. Okay? In either case, it's going to regulate what products are being formed. Okay? And it's a good regulation method. Okay? And lots of enzymes do this kind of method, and some biochemical pathways will have both options. Okay, you might have a competitor on one enzyme and an allosteric site on another enzyme, so you might go through the biochemical pathway and make one substrate, 2 proteins in, 3 proteins in, okay, and then you have this competitive that comes in, so the substrate can't be made anymore. Or you might have, right at the beginning, you might have an allosteric site that shuts it all down, so nothing can be made. And there's lots of ways to regulate enzymes.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
Okay, so there's a video showing you what I was just talking about. Okay. Now we're going to switch gears just a little bit to ATP, because we're moving from, energy, enzymes, that kind of thing, into photosynthesis. Okay? We're going to talk about biochemical pathways and the construction of ATP and NADPH and other molecules, energy-carrying molecules. Okay? But first we're going to talk a little bit about ATP. ATP is the currency of the cell, the main currency of the cell. This is the result of typically sun, photons, being converted into food sources. Okay? ATP is a very simple molecule because we already know a little bit about it. Okay? Adenosine triphosphate is what ATP stands for. And you've already seen this molecule before when we looked at DNA. The A is adenine. It's bound to a ribose sugar, and then it's bound to a phosphate group. Okay? Now, in the case of adenosine triphosphate, there are 3 phosphate groups. Everything else is very similar to DNA or the adenine nucleotide. Okay? So the structure of ATP really suits its role in being an energy carrier or the currency of the cell, and that is because it has sugar. Okay, I already told you it's a ribose sugar. It has adenine, that nucleotide, and then it has a chain of 3 phosphate groups. Now, most people would say, well, okay, I know where the energy is coming from. It's the sugar. But that's not the case. It's these three phosphate groups that is giving ATP molecule the energy that's needed. And what that is the phosphates are negatively charged. So you have a phosphate group hanging off the fifth carbon on the sugar of ATP. And then bound to that phosphate, you have another phosphate, which is negatively charged. And then bound to that, you have another phosphate, which is negatively charged. Now think about this. For those of you who have maybe played with magnets before, and where the magnet has a positive and negative side, and you've tried to stick two negatives together. They just are posed to, reject each other, pull apart. So ATP is already, when it forms, it's already going to fall apart. Okay? You can't store ATP molecules. We don't have the capability of storing it because it's not a stable molecule. But by having that negative bond to negative bond, okay, there's a lot of energy there, okay, that can be released and utilized in cell processes. So here you can see that molecule again. So you have adenine, again, off to the first carbon. The second carbon here is going to have an OH, which you recognize then that it's a ribose sugar. Okay, if it just has a hydrogen, then you would say, oh, it's deoxyribose. Off the fifth carbon, you have a phosphate group. So this piece right here is very similar to DNA. And then you have these negative bonds, okay, for the triphosphate. All right, so how does this work? Okay, that last phosphate will break off the ATP molecule. And so you get this. ATP becomes ADP, or adenosine diphosphate, inorganic phosphate plus energy. Right? And so how does ATP form? It's basically the exact opposite of this equation. You have adenosine diphosphate in solution, inorganic phosphates, okay? You add some energy and you can reform ATP. Now the cool thing about this piece is we have energy that's free coming to the planet that can drive the formation of ATP. That energy comes in the form of photons from the sun. Okay? And so this is the coolest part about the process is all life on the planet, okay, really is cyclic. The energy is not going anywhere. It's not being consumed. It's just being converted. But all the energy is free. Okay? The photons are free. So when we're talking about biological life, consumption of food, and things like that, that energy is free. Okay? As long as we're utilizing it from the sun. Okay? Now, often you will have coupled reactions. Reactions where an exergonic reaction is going to release energy, and that exergonic reaction, the energy released, is going to be utilized for the endergonic reaction that needs to take place. So you have these coupled reactions. Exergonic releases energy, endergonic brings it in. Okay? Now that really, okay, is a result of ATP, or the currency of the cell, being broken down into ADP. So endergonic reactions almost always happen when ATP is broken down. Okay? Now, when we look at this process, and we'll talk about this a lot more, there are times where more energy is being released than is absorbed, even by an endergonic reaction. And that energy is released into the atmosphere as heat. Okay? And we'll talk about this. Remember I talked about the 10% rule. Each time you convert energy, you only maintain 10% of that energy, because the rest is being released as waste or heat or different products that we cannot capture. Okay? And we'll come back to that when we talk about ecology. It's very important because that's really what drives ecosystems and why plants are so important to an ecosystem. And the more we cut down trees, the more we plow fields down and things like that, the less energy organisms have in that ecosystem. So we can look at this. Photosynthesis is just converting sunlight into ATP and then using that energy to make stored sugars or stored carbohydrates. Okay. Cellular respiration is taking those stored carbohydrates, stored sugars, okay, and breaking them down and converting it to ATP. So this is just a giant cycle. Okay. We're going to talk about photosynthesis soon, okay, and then we're going to talk about cellular respiration. And these two play a perfect role with each other. Remember, photosynthesis is taking carbon dioxide, water, and sun and making glucose, carbohydrates. Cellular respiration is taking carbohydrates and oxygen, okay, and making energy. What's the waste product of photosynthesis? Oxygen. What's the waste product of cellular respiration? Carbon dioxide. It's a giant cycle. It's perfect. It works great. Okay? And we'll talk about how these things work in the near future. Okay? So next time we're going to talk about photosynthesis. How is ATP formed? How are carbohydrates formed? Okay? And it can get a little bit confusing. So I highly suggest when it comes to photosynthesis, watch the videos multiple times, ask questions, those kind of things, because most of you know the very basics of photosynthesis, but we're going into detail on photosynthesis because it's really important to know some of the minute details, especially if you're into technology, how solar panels work, how the potential for hydrogen cars work, or hydro cars. All these different technologies, modern technologies, are going to come from things that we learn in biology. All right, till next time.